So with that, I'm going to turn to our introductions. I'm really excited to introduce Dr. Yaara Endevelt-Shapira, a research scientist at the Institute for Learning and Brain Sciences here at the UW. So she's right across the way. She earned her bachelor's in food engineering and biotechnology from Technician Israel. She completed her master's and PhD from the Department of Neurobiology at Weizmann Institute of Science in Israel, where she worked with Professor Noam Sobel on olfaction and social chemosignaling in autism spectrum disorders. She conducted postdoctoral work at the Center for Development in Social Neuroscience at Reichman University in Israel, where she worked with Dr. Ruth Feldman on social chemosignaling during early development, employing dual EEG to measure infant and adult interbrain synchrony. And with that, I'm going to stop sharing. And if I can find my button to stop sharing. I bet Dr. Endevelt-Shapira, if you just start sharing, it'll kick me off. I don't know where my button went. Oh, there it went. Okay, I found it. There we go. So welcome.
Okay, can you see
And
it?
we see it perfectly.
Okay,
Thank
great.
you.
Hi. So, wait, I need to. Okay, sorry, I just somehow there. So thank you so much for the introduction and for everyone for the opportunity to share my research here with you today. So as you just said, I'm Yaara Endevelt-Shapira. I'm a research scientist at the Institute for Learning and Brain Sciences at the University of Washington. I work here with professors Patricia Kuhl and Andrew Meltzoff on early social interactions, speech input, and language development. During my PhD, I worked on chemosignaling in autism spectrum disorder. My research in general examined how natural social signals influence human interactions and development. I study both speech, which is verbal cues, and nonverbal cues, chemosignals, and investigate how they shape behavior, physiology, and brain activity. So first, probably not all of you aware, what are chemosignals? Chemosignals in general are volatiles that are secreted from one individual to the surrounding and affect mood, behavior, hormonal state, physiological state, and even brain activity of other people that are exposed to these volatiles. It is well known that animals use chemosignals, or you may know it as pheromones, to communicate. In the recent years, there is a growing evidence for the existence of chemosignals in humans as well. One of the earliest reports suggesting the existence of chemosignaling in humans came from studies of menstrual cycle. Back in the 70s, a young student noticed that women that were living together in the dormitories appeared to synchronize their menstrual cycle. 20 years later, when that student already had her own lab, she published that what drives this effect is body odor. Specifically, women that were exposed to axillary sweat collected from other women during the day before ovulation experienced shorter menstrual cycles. So in other words, menstrual cycle of women that never met before became synchronized through exposure to body odor. Since then, many studies have explored chemosignals in human sweat. One of the most studied examples is anxiety sweat. So in general, anxiety sweat is collected from individuals that were in stressful situation. For example, while watching horror movie, during the first time of skydiving, or before an important exam. Most of the effects are reported mainly in women, but not in men. It was shown that the anxiety state of other individuals increased following exposure to anxiety sweat, and it also shown to influence how we perceive other people. For example, when women were exposed to fear body odor, they rated ambiguous faces as more fearful compared with when they were exposed to control odor. Other studies also found effects on cognitive performance and physiological responses. For example, exposure to fear sweat increased the human startle reflex, which is the automatic eye blink response that occurs when we hear a sudden, unexpected sound. So in addition to sweat, it was also found in our lab where I did my PhD that tears contain chemosignals. Very briefly, it was found that emotional tears obtained from women watching sad films decreased sexual arousal in men exposed to this odor alone compared to saline. Importantly, they were not aware what they're exposed to. It doesn't have like a perceived odor. So the decrease in arousal was demonstrated through decrease in testosterone levels, subjective feeling of sexual arousal, and decreased activity in brain regions that are related to arousal. So while most of what we know comes from lab studies where we actually expose the participants to odor, to jars containing body odors. But the question is, how body odors transmitted between us in everyday life? I mean, we used to do this every morning in our lab, but most people don't go around sniffing each other. So how do we sample chemosignals in real life? We hypothesized that a possible mechanism can be through a very routine gesture of handshaking. So in order to test this hypothesis, we invited participants to the lab. While they were waiting to their real experiments as they thought they were invited to, we had an experimenter introduce themselves and shake their hand. We measure if people sample their hand following the handshake. I'll let you see yourself.
Subject.
So here we shake the hand.
Subject, and right away a sample. Again.
We leave. So it was really strong, and we replicated many times. We used, I think, around almost 300 participants for this study. So I just showed you some examples of human chemosignaling. And there is a growing evidence for the importance of chemosignals in human social interaction and communication. I like to think of social chemosignals as a nonverbal language that is constantly used to convey information between us. And as individuals with autism spectrum disorders show difficulties in social interaction and communication, I thought it may be part of the altered social interactions of individuals with ASD could be related to altered chemosignal processing. So in order to test this hypothesis, we tested the influence of different chemosignals on both typically developed and ASD adults using several behavioral paradigms. I used both two different isolated chemosignals and natural fear sweat. Today, I'll show you only the fear sweat studies. So first, we collected fear sweat using my favorite paradigm from individuals that were doing their first time skydiving. We asked them to put pads under their armpits and collected them soon after the jump. In addition, we collected saliva samples before and after, just to verify that they were in stressful condition by measuring their cortisol levels. And they certainly were, like the cortisol level increased significantly. So first, we wanted to explore how the effect of fear sweat on both physiological and behavioral responses. So 20 individuals with ASD and 20 typically developed volunteers came to the lab. After the participants entered the room, and were sitting in front of a monitor, I placed a mask that both delivered the odor. I'll show you here, sorry, with the laser pointer. So this is the mask. And this mask both delivered the odor and also measured respiratory. And in addition, I placed EDA electrodes to measure skin conductance as a measure of autonomic arousal. So first they just watched a short video, and then they participated in the face task where they needed to rate ambiguous faces, the fearfulness of ambiguous faces. At the end of the experiment, I also asked all participants what was the difference between the two sessions. I mean, one session was with the fear sweat and one session was with control odor. Only 3 TD and 3 ASD participants have noticed that there was a difference in the odors. Actually, some of them were not aware that there was odor during the study. Like some of them came to me at the end of the study and asked me, hey, isn't that supposed to be an olfactory lab? Where was the odor? So it was, they were not consciously aware, most of them, of the odor condition. And yet what we found is that in the typically developed group, the GSR response, the EDA response, increased significantly compared to the control odor, while it didn't change in the ASD group. Here you can see the summation of the data. I observed a remarkable dissociation where the smell of fear significantly increased the autonomic arousal in TD, but not in ASD. The issue is that I didn't find behavioral effect. They didn't rate differently, not the ASD and not the TD, they didn't rate differently the faces. So I wanted to create a more realistic paradigm that will make the participants to actually behave instead of just sitting with electrodes in front of a monitor in the lab. For this purpose, we built Chris and Steve, our 2 realistic life-size mannequins. The participant entered a room where they stood in front of a monitor, and behind them stood the 2 mannequins. The subjects were asked to participate in a simple task in which they had to press on the button in the middle of the screen when they were ready, and then a blue star appeared either in the right or the left side of the screen. The participants were asked to press as quickly and as accurate they can in the place where the star appears. Before each of the tries, they were instructed to go to one of the mannequins to get an hint where the star is going to appear. They have been told that most of the time the mannequin says the truth. Yet the catch is that one mannequin is more reliable than the other. In fact, both gave the same number of correct hints. In addition, the participants didn't know that when they approach the mannequins, they emit odors. One emitted sport smell and the other fear smell. Because the mannequin's volume was also very low, participants tended to put their ears up close to the mannequin ear, like you see in B. This way, they put their nose in line with the emitted chemosignals. So basically, if they trusted one of the mannequins more than the other, we would expect to see quicker responses in the correct hints, since the participants already followed the hint and could move the cursor in the correct direction before the target appeared. Of course, the odors were counterbalanced, and I'll show you more in detail because I know it's maybe a bit complicated paradigm. So here you can see closer look of the front and the back views of the mannequins we built. In the back, we placed the jar with the odors, and the undetected odors were emitted from their nostrils. And also we had proximity sensors that they could sense when the participants approach them. So basically, I'll show you how it works. First, before each trial, it says, please approach Steve or Chris. And then when they are ready, after they got the hint, if they trusted the mannequin, they could already go to the side of what it said, like it's going to be in the left side of the screen. So they could already go there. But if it says, gave them wrong hint and they trusted it, they had to do all the way back. So basically what we measure is implicit measure of trust with the reaction times. If participants trusted a mannequin, this manifests in shorter reaction times to the correct hints or longer reaction times to the false hints. So what we found is that response time was faster for typically developed participants following the hints from the control mannequin, from the sports sweat. However, the SD participants trusted more the hints they obtained from the mannequin that emitted fear sweat. So overall, across also 2 different studies, we found that individuals with autism spectrum disorder display altered responses to social chemosignals, which emphasize the critical role of chemosignaling in human social interaction and communication. So I hope I convinced you that chemosignals are a meaningful channel of human social communication. These findings led me to ask how chemosignals affect social interactions during infancy. Before verbal language emerges, infants rely almost entirely on nonverbal cues across multiple sensory modalities to communicate with others and learn about the world around them. So what do we know about social chemosignals and specifically maternal body odor in human infants? We know that newborns orient toward the smell of their own amniotic fluid and their mother's breast odor. These early olfactory cues help guide newborns to the breast and begin feeding during the first feeding attempt. In addition, maternal body odor has been shown to reduce pain responses during routine ill-stick procedures in newborns. Other studies explore the effects of maternal body odor later in infancy. For example, it was shown that 4-month-old infants tend to look longer at faces compared to cars, and particularly in the eye region of the faces when they are exposed to maternal body odor compared to control. Another important role of maternal body odor is in emotional regulation in infants. In this study, 7-month-old infants were shown happy and fearful faces while their brain activity was recorded using EEG. The infants were divided into 3 groups. 1 that were exposed to their maternal body odor, 1 to stranger body odor, and 1 with no odor. And what we found is that both control groups showed the expected neural response to fearful faces, but this fear response was reduced in infants that were exposed to their own mother's body odor. So these findings suggest that infants process fear signals differently in the presence of their own mother body odor. And it supports the idea that maternal odor serves as a safety cue that helps regulate infants' emotional responses. So I was interested in exploring how maternal body odor influences infants' social behavior and neural responses during live social interactions. Probably, as all of you know, one of the fundamental features of social interactions is behavioral synchrony. Synchrony is the coordination of behavior between 2 people. It can happen at the same time, such as through mutual gaze or joint attention, or in sequence, such as turn-taking or imitation. Recent advances in hyperscanning methods, which is the simultaneous recording of brain activity, now allow us to study synchrony not only at the behavioral and physiological levels, but also at the neural level. So interbrain synchrony means that the brain activity of two people becomes coordinated during social interaction. You can think of it as two dancers moving through the same rhythm or two people being on the same wavelength. So the first evidence of adult-infant interbrain synchrony came from a hyperscanning dual EEG study by Vicky Leong and 2017. They found that when adults maintained direct eye contact while singing to the infant, their neural synchrony increased. Another study at around the same time, in 2018, showed that older children showed higher neural synchrony with their parents during cooperation game, but not with unfamiliar adults. Meaning that interbrain synchrony depends not only on the interaction itself and the content, like if it's cooperation or not, but also on the relationship between the two people. So these studies were published around the time I was finishing my PhD, and I was thinking about my next step. They made me wonder whether maternal body odor, which had previously been shown to increase infants' attention to faces and eyes, could also influence interbrain synchrony. At that time, I knew that Ruth Feldman had one of the first systems in Israel that allowed simultaneous EEG recording from infants and adults. So I reached out to her about a postdoctoral position and shared this idea with her. And luckily, she found this idea exciting as well. And just a few weeks later, I joined her lab to begin this project. So this study was published at Science Advances, and I'll share with you the main results. So for the experiments, mothers were provided with cotton t-shirts, and they were asked to wear them for 2 nights to collect their natural body odor. We asked them not to use deodorant, shampoo, or other scented products, and to avoid strong smelling foods, like avoid garlic or curry. The shirts were then stored in glass jars in the freezer until the experiment day. During the experiments, the infant and the adult interaction partner, which in this study was me, for 77 studies, I wore the EEG cup. The mother and both the unfamiliar women and the baby, all of them wore EEG cups to measure interbrain synchrony. The infants participated in 4 experimental conditions. So it participated in mother-infant back-to-back condition. The infant and mother were sitting back-to-back, watched soap bubbles, each or their own soap bubble machine, without direct interaction. This allowed us to measure neural synchrony without face-to-face communication. The other mother-infant condition was face-to-face. The mother and the infant interacted freely while we recorded their brain activity. The interactions were about 3 minutes each. In addition, in the stranger infant conditions, actually we had 2 conditions with the stranger infant. Once, in the presence of the mother body odor, of the t-shirt that she wore, we placed it on that shirt tray under the baby, and other, just a new blank t-shirt that no one wore before. I'll just let you see. I need to change from. Oops. Okay, sorry, I just need to switch it. It doesn't allow me. Hmm. Sorry, I'm trying to move back to the here. No. So this is me interacting with an infant. So I'm singing in Hebrew. And this baby was really cooperative. Not all of them. Out of 77 infants, we've completed the full paradigm with about 50 participants. Some of them didn't like the EEG cap, or some of them just didn't complete the full paradigm. So what we found. First, we compared interbrain synchrony between back-to-back and the face-to-face conditions, and found that mother-infant interbrain synchrony was higher during face-to-face interactions compared to moments of similar proximity, but without facial contact. I mean, you can see the result, and you can see in the heat map. At the x-axis are the infant's electrodes, the y-axis are the mother electrodes, and the most pronounced connection is between the right central region of the mothers and the right occipital-temporal region of the babies. Next, we compared neural synchrony during interactions with infant's own mother versus the unfamiliar women when no maternal body odor was present. And what we found, again, in the same region between the right central of the adult and the right central of the and the right occipitotemporal of the infant, that we found that infants had higher interbrain synchrony with their own mother compared to unfamiliar female. Finally, we compared the two infant-stranger interactions, one in the presence of maternal body odor and one with the control odor, and we found, again, at the same region between the right central of the stranger and the right occipitotemporal region of the infant, we found a higher interbrain synchrony during exposure to maternal body odor. Here we can see this region that I repeated over and over again between the right central of the adult and the right occipitotemporal of the baby. And as you can see, you can see here the comparison between the three conditions. And there was no difference in neural synchrony between the mother infant and the stranger infant interactions when infants were exposed to maternal body odor. So in other words, maternal body odor leveled out the difference in neural coupling or neural synchrony between the infant's own mother and unfamiliar adult. The interactions were also video recorded for later offline behavioral coding. We used two methods for coding. We used microcoding, where infants' gaze and affect were coded second by second during the interaction. I don't know if some of you are familiar with this system. It takes, like, for 3 minutes interaction, it takes about 3 hours of manual coding when we need to code each second and second. And the other method we used is global coding with the coding interactive behavior system developed by Ruth Feldman. The CIB, the global system, is broken down into parent, infant, and dyadic codes that are rated on 5-point scales. So what we found is that the presence of maternal body odor increased infants' visual attention, gaze, to faces. I mean, they looked longer at the stranger during the interaction when they were exposed to body odor compared to blank. It also increased their positive arousal. They smiled more and laughed more during the interaction when they were exposed to body odor, and also increased their safety approach behavior that was coding using the CAB. In general, this it composed of, for example, adult supportive presence, infant positive affect, infant initiation. And these are part of the scales: infant-led interaction, dyadic, adaptation regulation. I mean, so, and what we find really interesting is that while significant increases emerge in all of these three behaviors in the presence of maternal body odor, only the increase in visual attention was directly linked with the increase in infant-stranger interbrain synchrony. So it suggests a possible mechanistic link between infant social attention and neural synchrony. In general, as they looked more at the stranger during the body odor compared to the control condition, also the neural synchrony change score between the body odor and control condition was higher. So to sum this project, we found our findings suggest that maternal olfactory cues can help infants enter into new social groups, promote exploration of novel environments, and just broaden their overall social repertoire. So now the question is, what about fathers' body odor? So I'm very excited to show you this because it was published just about 3 weeks ago at Science Advances as well. And I can tell you in brief, yes, also fathers' body odor affect infants' interbrain synchrony and arousal. And now I'll show you more in details. So you can see this interaction between the stranger and the infant. I actually really like this video because you can see how the baby hold into the t-shirt and put it in his mouth and exploring it. So actually one of the things that motivated me to explore fathers is that while I was writing our paper on maternal body odor, I came across a fascinating anthropological study describing a traditional ceremony in which newborns were introduced to their fathers for the first time. During the ceremony, the fathers placed their hand under their armpit and transferred their scent to the infant's head. The idea that body odor could play a role in creating early father-infant connection really inspired me to explore fathers' body odors as well. So the paradigm was very similar to the mother body odor study. Again, we collected fathers' natural body odor by asking them to wear a new t-shirt. We provided them for 2 nights. And during the study, both the infant and the adult were EEG sensors, so we could record their brain activity at the same time. And in this study, we attempted to complete the back-to-back condition. However, many infants did not complete this baseline paradigm because the infants were distressed, they were crying, or limited engagement with the screen. Oh, another difference, in the first study, we used actual bubble machine. It was pretty messy after. So we decided to change it in this study to just watching short 2 minutes bubble on the screen, but apparently it was also less engaging. And so we ended up having only 3 paradigms to compare: the father-infant face-to-face, and the 2 stranger infant conditions. So first, we compared the interbrain synchrony during the interactions with on father compared to the unfamiliar father with no paternal body odor present. And interbrain synchrony was higher between infants and their own father compared to the unfamiliar male. And you can see it in a bit different regions than we previously saw in the mothers. Actually, the infants regions are really similar. It's in the right temporal, occipitotemporal regions, but the father, instead of the right central, it was on the left central, which is interesting. Next, we compared the two infant-stranger interactions and found that exposure to paternal body odor increased neural synchrony between infants and the unfamiliar male. And again, it was in the same region between this left central and the right temporal region of the baby. We also measured the free behavioral construct that we used with the mothers. And we found that exposure to father's body odor increased infants' positive arousal during the interaction, but didn't change infant visual attention or safety engagement, as we saw in the mother study. Moreover, I find this result really interesting. And fathers that were more involved in daily childcare showed stronger effects of paternal body odor on infant brain synchrony. It means that more sensory association, that this sensory association may become stronger with repeated caregiving experiences. So our findings show that infants, even during early infancy, babies' brains are more synchronized with their own father during social interactions than with unfamiliar adults. Moreover, infants are sensitive to their father's natural body odor, which enhances interbrain synchrony and positive arousal during interaction with the unfamiliar adult. As fathers become more involved in everyday caregiving, this influence becomes even stronger, suggests that these repeated everyday experiences shape how infants respond to their father's unique sensory cues. So moving forward, we want to understand how parental scent, parental body odor contributes to early social development. Does a familiar caregiver's odor help infants during moments of separation? Or does it influence social engagement and interbrain synchrony also across different developmental populations? Personally, I'm really interested exploring it in infants at elevated likelihood of ASD. And also, how does this sensory influence extend behind parents to other important caregivers? So all of these questions still need to be addressed. So far we have talked about parental body odors. In the next studies I explored how maternal behavior is related to interbrain synchrony and how these early interactions are linked to later development. So first, I examined interbrain synchrony association with two well-studied maternal behavior orientations, maternal sensitivity and intrusiveness. We measure interbrain synchrony during face-to-face interactions between mothers and their infants and video recorded the interactions. So sensitive caregiving in general means that the mother noticed and responds to her baby's signals in the moment and adjusting her behavior based on the baby's needs and emotional state. This kind of back and forth interactions help support development of the child's social and emotional abilities, In contrast, intrusive caregiving occurs when parents' actions are driven more by their own agenda rather than the baby's cues, and leading to interactions that may be overwhelming to the infant. So decades of research have shown that these different patterns of early interactions are associated with different developmental outcomes, with sensitive caregiving supporting positive social emotional development and intrusive caregiving linked to less favorable outcomes. Important to say in this study, actually, it wasn't that we had intrusive mothers or sensitive mothers. We used the same interaction, same mothers, and we coded them for both intrusive and sensitive caregiving. So what we found is that higher maternal sensitivity and lower intrusiveness are associated with higher mother-infant interparent synchrony. We think that the quality of early parent-infant interactions may shape the development of the social brain, and this could help explain the long-term effects of children's behavior and development. So as maternal sensitivity has been shown to be associated with both positive social emotional outcomes and also language development, I was motivated to directly study the relationship between early parent-infant interactions, including maternal sensitivity, and later language developments, which led me to join to the Institute of Learning and Brain Sciences at the University of Washington. Previous studies have highlighted the importance of social interactions and speech input for child language development. Although infants begin to engaging in this interaction by the 3rd month of life, most of the links that were published between speech input, maternal sensitivity, were published later, and almost there are no studies showing these links between this early development window at 3 months old and later outcomes. And I was interested to explore this early period of early development window at 3 months old. So we designed a longitudinal study. It included 3 parts. First, to directly assess mother-infant interactions, mother and infants came to the lab and to participate in free play interaction session. Mothers were instructed to interact naturally with their infants. Second, a week after the lab visit, families completed Lina recordings in their home environments to assess infants' everyday language input. The Lina system is capture naturalistic language environments by recording children's everyday auditory experiences over 2 days. It can be stored up to 16 hours of recordings, and they can place it, as you can see here, in a child vest that we also provide to the parents. The third part, so these two, the mother-infant interaction and the Lina recordings, were at 3 months old. The next part is that we followed language development between 18 to 30 months old, using the parents completed this assessment using the MacArthur Bates Communicative Development, CDI. And eventually we used vocabulary percentile scores. Parents check the words the child can produce on a word list with a maximum number of 680 words. So this is, there are different measures in the CDI, but we focus on the vocabulary scores. For speech input, we focused on 2 aspects of parental speech input. infant-directed speech, and conversational turns. When I joined iLabs, my own children were already in first grade and pre-K. And then looking back, I realized that no one ever told me when they were infants how important it is the way we talk to our babies. So it made me really curious. And I went back through old family videos to see whether I had naturally used infant-directed speech with my own children. Thankfully, I had. Like many parents, I found myself speaking in higher pitch, exaggerated intonation, and slower, more expressive speech without even being aware of it. That's actually, I think it's one of the fascinating things about infant-directed speech, that it appears to be so natural and found across cultures and languages and produced by caregivers around the world. So I can show you here two short clips of me talking to my children, one with my daughter and one with my son. Even though the language is Hebrew, I think you will be able to hear the melody of infant-directed speech.
בוקר טוב, ישנת הרבה שעות?
And with my daughter, she's 11 now.
איזה חמודה, איזה חמודה, יפה כזאת, את כזאת יפה.
So we found that maternal sensitivity at 3 months and infant-directed speech and conversational turns, importantly during one-on-one interactions at the home environment, were positively correlated with productive language scores. And why I'm saying importantly during one-on-one interaction, because we also wanted to check what's going on in group context compared to one-on-one context. And we found that these associations are specific to one-on-one. There is something about infants' interest and attention that the most of the support and the correlations were shown only during the one-on-one interactions, both for the conversational turns and both to the parentese or infant-directed speech. To further explore the long-term effects of early social interactions, when participants from the same group reached 5 years old, they also came to the lab and completed a clinical evaluation of language fundamental preschool. It's so-called the SELF-P, a comprehensive individually administrative assessment of language skills. And we found, out of 40 participants that started this study when they were 3 months old, we were able to reach 32 that completed this assessment at 5 years old. And we found significant associations between early one-on-one interactions, including infant-directed speech and conversational turns, and phonological awareness at age 5. I'm currently finalizing these findings and preparing the manuscript for this. So these findings led me to investigate the neural and physiological mechanisms underlying language learning. We know that during early social interactions, So what I hypothesized is that one of the mechanisms that infant caregiver interactions during this early period support language development is increased infant attention to speech. In infants, attention and orienting responses are associated with heart rate declaration. So in the same 3-month-old cohort, we also recorded during another interaction, infants' ECG, and allowing us to measure infants' heart rate. And I found that higher maternal infant-directed speech and conversational terms and the home environment are associated with lower infants' heart rate measured during mother-infant interactions at the lab when they were 3 months old. Moreover, it was the vocabulary percentile scores at 30 months old during the third year of life was associated with heart rate. Lower heart rates were associated with higher language development. It further supports the hypothesis that infant social attention to speech during this early development window may play a role in advancing language development. Obviously, I would be happy to try to have bigger sample size, and I think we ended up with 28 participants for this part due to some technical issues, but I think it's really interesting if we could identify as early as 3 months old using quick physiological measure. We just need 3 minutes interaction for this. If you can detect populations that may be at risk for later language development. So in addition to this physiological measure, I also hypothesized that one pathway through which early parent-infant interactions extract their long-term effects on language development may be through increased interbrain synchrony, which in turn supports social brain maturation. As I showed you a few slides ago, I found that maternal sensitivity is associated with interbrain synchrony, and that it's known that sensitivity is also play an important role in language development. So to test this hypothesis, we conducted a longitudinal study here at iLabs using multiple methods. We used EEG recordings for mother-infant and also father-infant interactions, but I'm still analyzing this. And we used behavioral coding and recording of infants' auditory home environment using the Lina system and following their language development as well. So we measure interbrain synchrony during mother-infant interactions at 3 months old and found 5 significant connections. And we explore then whether these connections that are found to be significant are also associated with vocabulary scores at 18 months. 2 of these, of the 5 significant interbrain connections were found to be correlated with vocabulary percentile scores, which is really exciting, honestly. I mean, we found it right temporal to right temporal, interbrain synchrony, and the right central to right temporal interbrain synchrony are associated with later language development. And also the mother-infant right temporal to right temporal connection was significantly associated with maternal sensitivity that was measured during the same interaction. And even mediation analysis revealed that interbrain synchrony partially mediated the link between maternal sensitivity and later language development. It is interesting, actually, that among the 5 significant mother-infant connections that were identified during the face-to-face interaction, this was the only connection that was linked to both maternal sensitivity and later language outcomes. The right temporal region is a keynote of the social brain, which supports processing of socially meaningful signals. And so this finding, together with previous research showing the importance of early social interactions for language learning, and support the idea that social interactions help to gate early language development. And mother-infant early interbrain synchrony can also serve as a marker of the quality of early social connections that support language learning and long-term developmental outcome. So to conclude, our findings suggest that early communication is shaped by the quality of social interactions and the broader social and sensory environment in which learning takes place. The coordination between infants and their caregiving through voice, interaction, and body odors may provide an important foundation for social and language development. Moving forward, I'm interested in understanding how this process also unfold across different developmental trajectories, including children with diverse communication profiles. I also want to better understand the role of fathers and other caregivers, and to identify if there are specific features of speech input that are more strongly related to interbrain synchrony and later language development, and specifically if they are different between different populations. So with that, I would like to thank you for attending and listening, and to thank my mentors, Noam Sobel, Ruth Feldman, Patricia Kuhl, and Andy Meltsov.